Hello, I'm Jean Riddell, and this is a presentation on immunology. The target audience is medical laboratory technician or medical laboratory scientist students. The reference book is Modern Blood Banking and Transfusion Practices 6th edition by Denise M. Harmoning. This is Chapter 3. Please consider subscribing to my channel, liking and commenting, and sharing the presentations with fellow students. Please comment with other topics that you are interested in. The objectives are to be able to differentiate the types of immunity, to be able to define terms and apply them to blood bank activities, differentiate the types of antibodies and their functions, understand the types of antibodies significant in the blood bank, understand the effect of temperature on antibody reactions, differentiate the types of complement activation, differentiate between polyclonal and monoclonal antibodies. Terms and definitions. Immunity, the process by which a host organism protects itself from attacks by external and internal agents, protection from non-self and abnormal self elements, which are controlled at different levels. Cellular immunity, an immune response that does not rely on the production of antibodies. Rather, cell-mediated immunity is the activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen. Humoral immunity. Humoral immunity is also called antibody-mediated immunity. This physiological mechanism protects the body from pathogens and foreign substances in extracellular fluids and is part of both the innate and adaptive immune systems. It involves a humoral immune response that occurs in two stages, primary and secondary. The primary phase is set into motion upon the body's first contact with an antigen, surface protein found on a pathogen membrane, for example. The secondary phase describes the immune system reaction to subsequent contact with the same antigen. Immunoglobulin, also known as antibodies, one of the family of closely related but not identical proteins that are capable of acting as antibodies. There are 5 types: IgA, IgD, IgE, IgG, and IgM. Alloantibodies, an antibody produced against the antigens of another person. In blood banking, specifically against non-self antigens on red blood cells. Autoantibodies, an antibody produced against the antigens of your own cells. In blood banking, specifically against self-antigens on red blood cells. Innate immune response, less complicated and more primitive system that does not function in a specific way. It recognizes complex repeating patterns present in common invading organisms, and this is your first response to exposure to a potential pathogen. Acquired immunity, an advanced system that relies on the formation of specific antigen-antibody complexes and specific cellular responses. Memory of previous exposure to pathogens acquired through infection or immunization. Phagocytes. These are cells that engulf foreign pathogens, antigens, to remove them from circulation. Part of the innate immune system. These are polymorphonuclear cells, neutrophils, basophils, and eosinophils, monocytes, macrophages, and dendritic cells, and they act as antigen-presenting cells. Opsonin, a factor such as an antibody or complement that coat pathogens or foreign cells to facilitate phagocytosis. Process is called opsonization. Complement, a series of proteins in the circulation that when subsequently activated cause disruption of bacterial and other cell membranes. Activation occurs by different pathways. Activation of complement by antibodies to foreign red blood cells can result in hemolysis of red blood cells. Antigen, an epitope or antigenic determinant, a substance recognized by the immune system as being foreign, which can cause an immune response. In blood banking, antigens on the surface of red blood cells that can react with antibodies. An immunogen. Serology, the study of antigen-antibody reactions. Plasma cell, a B lymphocyte that has been stimulated by an antigen and differentiates into an antibody-producing cell, produces an antibody against one specific antigen. Major histocompatibility complex, or MHC, HLA antigens. These are used by antigen-presenting cells to present foreign antigens to other white blood cells. Antigen-presenting cells. Macrophages, neutrophils, and some B cells can act as antigen-presenting cells. They phagocytize foreign antigens, process it, then present the antigen to the MHC, on the MHC to immune cells. This stimulates the immune system. Classical complement cascade. A complement cascade initiated by IgG or IgM antibody antigen complexes. Alternate complement cascade. Complement cascade activated by surface contact with complex molecules and artificial surfaces without acquired immunity. Lectin complement cascade. Activation by the attachment of mannose binding lectin to microbes. Membrane attack complex. This is C5b6789, the final activation of all complement cascades, which causes cell lysis. In blood bank, this leads to hemolysis. Polyclonal antibody, a type of reagent antisera. These have heterogeneous antibodies. Multiple antibodies to the same antigen but different epitopes of the antigen. They are naturally produced antibodies, are also polyclonal. Monoclonal antibodies, a type of reagent antisera, which has homogeneous antibodies, a single antibody to one specific epitope, and is produced by hybridoma technology. Anti-human globulin, a reagent antibody that reacts to human antibodies, so an antibody to an antibody, enhances detection of IgG antibodies. Unexpected antibody, an antibody that should not be present under normal circumstances. It is not naturally occurring, produced after exposure to an antigen. Naturally occurring antibodies, antibodies that are expected to be present without exposure to the corresponding antigen. No known exposure to the antigen is required for you to produce these. ABO antibodies such as anti-A, anti-B, and anti-A-B are naturally occurring antibodies. Affinity, the strength of a single antigen-antibody bond produced by the summation of attractiveness and repulsive forces. Avidity, the binding strength of a multivalent antigen with antisera produced in an immunized individual, a combination of affinities of the polyclonal antibodies. Specificity, the affinity of an antibody and antigen against which it is directed, how specific it is to a certain antigen. Immunology. The immune system is a highly complex system that includes organs, cells, tissues, and chemical mediators that work together to defend a person against foreign substances, pathogens, or cells from another person. So we can divide the immune response two different ways. First, cellular and humoral. Second, innate and acquired. Cellular immunity is innate, but there is more to innate than just cellular immunity. Humoral immunity is also acquired immunity. Cellular immunity is one of the two major divisions of immunity. It is mediated by immune cells such as macrophages, T cells, and dendritic cells. Effector molecules called lymphokines are powerful molecules that act as a means of communicating the needs for the immune system to be activated. These are cytokines and chemokines. Humoral immunity is also known as antibody-mediated immunity. B cells become activated by encountering an antigen or being presented an antigen by an antigen-presenting cell. They can then differentiate into memory cells or plasma cells. The plasma cells produce antibodies to the antigen to destroy the current infection. The memory cells are ready if you become infected a second time. These antibodies are specific to a certain antigen. Natural immunity is acquired by infection, and artificial immunity is produced by vaccination. Both of these are active immunity. Innate immune system allows you to protect yourself from random rather than specific threats. It is a natural protection and not acquired. It is not as complicated as acquired immunity. It is not specific and stands ready to protect you even if you have not come in contact with a pathogen in the past. It consists of physical barriers, biochemical effectors, immune cells, skin, enzymes, tears, stomach acid, and phagocytes, for example. Acquired immunity is more advanced. Exposure to an antigen generates immunity. It is specific to one antigen only. Plasma cells are generated that produce antibodies, and resistance to pathogens that have been encountered so that you can fight off the infection if you encounter it again. This chart shows you the characteristics of the different types of immunoglobulins, which are classified by their molecular structure of their heavy chains. So we have IgA, IgD, IgE, IgG, and IgM. The IgG and IgM and IgA are the ones that are most clinically significant in blood banking. IgG and IgM can both activate complement, which can be dangerous when you're talking about activating it on a red blood cell. And IgG can cross the placenta, which can cause problems and hemolytic disease of the fetus and newborn. IgA antibodies are a dimer, and it's a problem when you have people with IgA deficiency. IgD is a B cell receptor. IgE is found elevated in allergies and parasites. IgG is a problem with transfusion reactions, hemolytic disease of the fetus and newborn, and they react at 37 degrees Celsius. IgM is a pentamer. It reacts at immediate spin. These are also the ABO antibodies, which if you don't select the right blood type can cause deadly transfusion reactions. Complement is a complex group of cell membrane proteins that have many functions in the immune response. They cause the direct lysis of cells, bacteria, and envelope viruses. They perform opsonization to facilitate phagocytosis. They get the inflammatory response going, and they are activated via a cascade. The classical complement pathway, the activation of this pathway is initiated when an antibody attaches to an antigen. This can be IgG or IgM antibodies that activate these. This is part of the acquired immune system. In blood banking, IgG or IgM antibodies can bind to foreign red blood cells and activate complement, causing the lysis of transfused red blood cells. And of course, we want to avoid this in blood banking. The alternate complement pathway is activated by surface contacts with complex molecules and artificial surfaces such as dialysis membranes and dextran polymers. We do not want this to be activated to destroy red blood cells. The lectin complement pathway is activated by attachment of mannose binding lectin to microbes. The membrane attack complex is the end of each of the different pathways. So all of them end in a membrane attack complex, which is C5b6789, and cause the lysis of the cell. And while that's good with the bacteria, that's bad if it's red blood cells. Immune antibodies in blood bank. These are caused by either by transfusion or pregnancy, both which can expose you to foreign red blood cells. IgG antibodies are those that react after incubation of testing at 37 degrees Celsius, and it requires the addition of an anti-human globulin reagent to get it to clump in the tube. Examples are the Rh antibodies, KID, Duffy, Kell, and S. Laboratory reagents used in the blood bank can be either monoclonal or polyclonal antibodies that we use to type patient red blood cells or to detect antibodies to red blood cells. In the blood bank laboratory, the gold standard method is tube testing. This detects hemagglutination and hemolysis. Precipitation and inhibition are not common testing methods in the blood bank. Other methods of blood typing are gel and solid phage, which are automated methods that you will have the opportunity to work with in your clinical rotations. The red cell agglutination reactions. This happens in two stages of hemagglutination. First, there's sensitization. This is when the antibodies attach to the red blood cells. And then there is lattice formation when the sensitized red blood cells link together to form a clump. The volume of antigen and antibody added to a test must be consistent to avoid false negative reaction. A prozone is when lattice formation does not occur because the cells become so coated with antibodies that they don't cross-link and agglutinate. In the postzone, there are too many antigens and the antibodies can't cross-link for agglutination to occur. The zone of equivalence is when you have the appropriate amount of antigen and antibody to form agglutination. This image shows you the effects of having the antigen-antibody ratios in the correct and incorrect ratios and how this can cause false negative reactions by preventing lattice formation. In the first example of the prozone, you can see that there's so many antibodies that they coat every single antigen on each red blood cells, and then you don't get the lattice formation. So the cells are still free floating. In the zone of equivalence, you have cross-linking occurring and you get this clump of cells. In the antigen excess, you get what's called a post zone, so you don't have enough antibodies compared to your antigens to cause cross-linking to occur and no lattice formation. Temperature is important in blood bank testing. IgM antibodies react best at immediate spin, which is room temperature or colder. IgG antibodies react best at 37 degrees Celsius incubation with anti-human globulin added. We also use enhancement media, which works to reduce the zeta potential that keeps red blood cells separated. We have low ionic strength saline, polyethylene glycol or PEG, and we have proteolytic enzymes, trypsin, physin, and bromelain, which have different effects for different antibodies reactivity. Questions. Question 1. What type of immunity is not specific? A, acquired, B, artificial, C, innate, D, antibody. The answer is C, innate. A person who had the flu and made antibodies has what sort of immunity? A, natural innate, B, natural acquired, C, artificial acquired, D, artificial innate. The answer is B, natural acquired immunity. Question 3. Humoral immunity is regulated by A, B cells, B, T cells, C, macrophages, D, neutrophils. The answer is A, B cells. Question B. An antibody that reacts with donated red blood cells but not your own is called what? A, autoantibody, B, immunoglobulin, C, alloantibodies, D, IgE antibodies. The answer is C, alloantibodies. Question 5. Unexpected red blood cell antibodies that cause transfusion reactions and hemolytic disease of the fetus and newborn are A, IgA, B, IgM, C, IgG, D, IgE. The answer is C, IgG antibodies. Question 6. Antibodies that we expect to find in some of our plasma that react with incompatible blood types at immediate spin. A, IgA, B, IgM, C, IgG, D, IgE. The answer is B, IgM. If an unexpected antibody attaches to a red blood cell, what type of complement activation would this cause? A. Transverse pathway, B. Lectin pathway, C. Alternate pathway, or D. Classical pathway. The answer is D. Classical pathway. Question 8. A monoclonal antibody is created by A. Injecting a rabbit with human blood, B. Hybridoma technology, C. Transfusing a human with human red blood cells, or D. Digestibility technology. The answer is B. Hybridoma technology. Question 9. Cell-mediated immunity involves A. T cells, B. B cells, C. Plasma cells, D. complement. The answer is A. T cells. Question 10. A process that coats cells or antigens with complement or antibodies to enhance phagocytosis is A. Lattice, B. Opsonization, C. Hybridization, D. Hemagglutination. The answer is B. Opsonization. For more questions, you can find me at Kahoot if you search my name, Jean Riddell, without a space, and you'll find urinalysis, immunology, and immunohematology questions as well as phlebotomy. Thank you, and please like and share.